David Lagares. I'm an assistant professor of medicine at Harvard Medical School. I'm also the co-founder of Media Therapeutics and Xenon Biotech and the organizer of the next Tissue Repair and Fibrosis meeting in March 2023 in Banff National Park. So this meeting will focus on the pathogenesis of human fibrotic diseases, which are characterized by pathologic scarring of the organs in response to tissue injury. This fibrosis will ultimately lead to organ failure. I really want to push people to think about three emerging questions in the field of fibrosis and regeneration. And these questions revolve around mechanisms driving the progression and perpetuation of organ fibrosis, and we will focus on aging, mechanobiology, and epigenetic modifications. We will also focus on fibroblast heterogeneity. These are the effector cells in tissue fibrosis, and we will focus in particular on novel technologies that are allowing us or enabling us to identify novel subsets of fibroblasts and functions of these fibroblasts in tissue repair and fibrosis. Ultimately, the main goal of this meeting is to think about novel therapeutic approaches to resolve fibrosis and regenerate the organs. And we're going to be discussing not only with academic scientists and physician scientists in academia, but also industry partners, novel approaches to develop antifibrotics that lead to the resolution of fibrosis, which is a novel class of drugs that haven't been investigated yet. I'm really interested in the discussion between academics and industry partners, and the ultimate goal is to not only understand the mechanisms of driving fibrosis and fibrosis resolution, but more importantly, think about how to translate this into the clinic. We're going to be thinking about major clinical challenges translating antifibrotic therapy from the bench to the bedside. We're going to be focusing on fibrosis biomarkers that need to be discovered, clinical endpoints and safety endpoints when running clinical trials, which are going to be needed when thinking about translating these therapies into patients. And for that, we're going to be talking about technologies that enable us to study epigenetic modifications and also mechanobiology and how bioengineered tools can help us advance these knowledge gaps in the field. Again, it's going to be an overlap between cell biology and drug development. So from academics, we're going to be touching base on stem cell biology, epithelial cell biology, fibrous biology, and immune system. So this is going to be in the context of tissue repair and resolution. Not only cell biologists, but also people developing technologies such as single-cell RNA-seq and CAR T cells. These kind of technologies that will definitely help us reverse fibrosis and regenerate the organ. Postdocs and PhD students are welcome to submit their work in the form of abstracts. We will select short talks for presentation in the meeting. We will also have career development seminars and workshops, so you will have the opportunity to interact with established leaders in the field. Organizing this meeting has been a tremendous joy for me. The level of science is pretty high, so I got to interact with leaders in the field. So finally, I would like to acknowledge my co-organizers, Professor Boris Heinz from the University of Toronto, and Karim Bustani from Boehringer Ingelheim for helping me putting this terrific program together. And of course, the SAB of the Keystone Fibrosis meeting for giving me the opportunity to organize this important conference for the field.